In this experiment, you will observe photosynthesis and cellular respiration by looking at leaf discs float and sink. For this experiment, you will need 2 clear plastic or glass containers, a light source, a hole puncher, and leaves. Spinach leaves work well. You will also need a plastic syringe that is 10 cc or more, with no needle. Next, you need the kitchen item sodium bicarbonate, which is baking soda, and liquid soap, such as dish soap. Finally, you need some means of keeping time, as you need to collect data at set time intervals. First, label your 2 plastic containers experimental and control group. Next, you need to pour 300 milliliters of water into the container labeled experimental. This is approximately 1 1/4 cups if you don't have a measuring cup that shows milliliters. Next, add 1/8 teaspoon sodium bicarbonate to the container and stir until it is completely dissolved. Next, add 2 drops of solution and stir. The soap will help make the leaf discs absorb the solution. You do not want suds to form, so stir gently. Now that the solution is ready, use the hole puncher to cut out 10 discs from the leaf. Avoid major veins in the leaf. Next, place the leaf discs in the syringe. Place the plunger back on and carefully depress it. Do not crush the discs, as you will have to cut out new discs. Next, you will draw fluid into the syringe. Make sure you draw fluid from the container labeled experimental group. Hold the vacuum for about 10 seconds. In this next step, we are trying to force the liquid into the leaf disc by creating a vacuum and replacing the air-filled spaces with the sodium bicarbonate solution. When the leaf discs are full of liquid, they will sink. Most likely, you will need to repeat this step several times until all the discs sink. Finally, you need to get the discs out of the syringe and into the experimental container. Now, you need to set up your control group. First, pour 300 milliliters, approximately 1 1/4 cups water, into the container labeled control group. Again, only add 2 drops of soap and gently stir to avoid suds. Do not add sodium bicarbonate to this solution. Cut out 10 more leaf discs and repeat the same procedure as for the experimental group, but make sure to use the control group solution. We are now ready to start the experiment. First, we want to make sure the discs are not stuck to the container wall. So swirl the containers, but make sure the discs still sink to the bottom. We will first see what happens when we expose the leaf discs to light. We should expect to see the discs start floating. Start your timer, and after 1 minute, record how many discs are floating. Record for each minute for 20 minutes. After 20 minutes, we are going to eliminate the light source. Record the number of discs floating every minute for another 20 minutes. You have now collected data for 40 minutes, and it is time for you to analyze the data. Answer the questions and graph your data in the lab manual to get to a conclusion from this experiment.